Hi, I'm Peter Libby from the Harvard Medical School and the Brigham and Women's Hospital, and it's my honor to participate as the American lecturer in this year's virtual Journée Européenne de Cardiologie de la Société Française de Cardiologie. I've been asked to speak in 15 minutes about inflammation and atherosclerosis, and I thought that I would take you on a bit of a translational journey, starting from the basic science bench into large-scale clinical trials. These are my competing interests. Well, we now understand that inflammation is part of atherosclerosis from the very beginning through the long phase of progression of this disease, which may be clinically silent or stable, and up to and including the thrombotic complications of atherosclerosis that bring the patients to our attention as clinicians most dramatically. I spent a great deal of time working on the various phases of atherosclerosis, dissecting inflammation. And in particular, we worked out the cell biology and molecular biology of instability of the atherosclerotic plaque as related to inflammation. We see the blue lymphocytes and the yellow foam cells derived from macrophages in the inflamed plaque. And we have worked out the pathways by which the T lymphocytes can secrete mediators that impede the ability of the smooth muscle cells to manufacture new collagen that is responsible for the integrity of the plaque's fibrous cap. We also worked out in considerable detail the inflammatory signaling that causes the overexpression of a series of proteolytic enzymes, the interstitial collagenases, that are specialized in breaking down collagen. Since it's collagen that lends strength to the plaque's fibrous cap, when we have inflammation in the intima, it's a perfect storm with decreased synthesis and increased breakdown of collagen, weakening the fibrous cap and rendering it susceptible to thrombosis. We've also, with my research partners, Matthias Narendorf and Phil Psursky, invoked a connection between the central nervous system and the bone marrow and even the spleen in the cardiovascular continuum, because leukopoiesis that is controlled by alarm signals and sympathetic drive can increase hematopoiesis and cause leukocytes to enter the plaque, and also can activate those that are already present, leading to a vicious circle. We have a number of traditional risk factors that drive inflammation, such as smoking, dyslipidemia, hypertension, and dysglycemia. And we know also that lifestyle is very important. Evidence is increasing that lifestyle changes can be anti-inflammatory. There are also emerging risk factors that provoke inflammation, such as the triglyceride-rich lipoproteins, adipose tissue, particularly visceral or ectopic adipose tissue, infectious agents, and, as I will talk about in a moment, clonal hematopoiesis. Well, it took a Google Scholar a few days ago 0.07 seconds to find over 1.5 million hits for inflammation in atherosclerosis. But the challenge is to translate the inflammation biology in atherosclerosis to the clinic. So I've set out the task of answering this question: can targeted anti-inflammatory therapy improve cardiovascular outcomes in humans? We focus first on interleukin-1 beta, and I refer you to this review for all of the reasons why. Of course, interleukin-1 beta can elicit from the cells that are key participants in atherosclerosis a number of functions that are related to atherosclerosis and its complications. I hypothesized in print a role for interleukin-1 in atherosclerosis in 1986. So my laboratory has been working on this for a very long time indeed. Now, like many proteins that are important in biological control, interleukin-1 is first synthesized as an inactive precursor and requires a limited proteolytic nick in order to attain its biological activity. This is very similar to angiotensin 1 being cleaved by angiotensin converting enzyme to active vasoconstrictor hormone. The same kind of scheme controls the activity of interleukin 1 beta. I'm not going to take you through the years of basic science work in my laboratory. The first demonstration in 1986 that vascular cells could make interleukin 1 beta, the ability of interleukin 1 to induce its own gene expression, providing the lymphification loop, the finding that IL-1 can induce interleukin 6, this is a logarithmic expansion, very strongly, and we found in the atherosclerotic plaque the machinery, the enzyme that causes the maturation of pro-IL-1 beta to the active form. We know that after these two amplification loops of autoinduction and induction of interleukin 6, that the interleukin 6 can cause the hepatocyte to change its program of protein synthesis to favor the acute phase response, including fibrinogen and plasminogen activator inhibitor, which together can promote accumulation of thrombus. We know that the enzyme that cleaves pro-interleukin 1 beta is part of a supramolecular structure known as the inflammasome, which responds to danger signals, including cholesterol crystals and other plaque-related stimuli, to activate caspase-1 to provoke the maturation of pro-IL-1 beta to the active principle. This, by the way, is in stained glass, a long axis view of the NLRP3 inflammasome superimposed over an atherosclerotic blood vessel. And this is the ribbon structure of interleukin 1 beta. Now, I talked about clonal hematopoiesis as a novel risk factor. If you'd like to read about it in detail, I refer you to this recent publication, At the Crossroads of Aging, Cardiovascular Disease, and Cancer. Mutations in stem cells in the bone marrow can give rise to cells that have a defect in a driver gene of leukemia that causes a clonal expansion that one can see in peripheral blood. And while accumulation of these mutations can lead to acute leukemia, the road to acute leukemia is actually paved by cardiovascular disease. It's a very recent finding. It's only a handful of the 40 or 50 genes that drive leukemia that are associated with these clones, which accumulate with age, so that by the time you're 70 years old, you have at least a 10% chance of having one of these clones. And if we look at the cardiovascular risk in people who have this condition known as clonal hematopoiesis of indeterminate potential, it's as strong as most traditional risk factors, except for diabetes and age. When we modeled clonal hematopoiesis in the laboratory, we found that the leukocytes from these mutant animals, when stimulated with low-density lipoprotein, LDL, overexpress interleukin 1 beta and interleukin 6, the very pathway that we have implicated in atherogenesis for many years. And looking in humans, in this very recently published paper from my collaborators in Nature, you see that humans have increased expression of interleukin 1 beta when they have one of these mutations, somatic mutations that they've acquired in bone marrow stem cells. So armed with all of the biological plausibility, together with Dr. Ridker, I designed and helped to lead a study where we neutralized interleukin 1 beta with a monoclonal antibody. This was, of course, the CANTOS trial, where we were able to reduce inflammation as gauged by lowering C-reactive protein without modifying atherogenic lipoproteins. We found a 15% reduction in the pre-specified primary endpoint, which is numerically exactly the same as one saw in Fourier with evolocumab and in Odyssey with alirocumab. But this is independent of changes in lipids. We paid a price in terms of inhibition of host defenses, there was a small but significant increase in fatal infections. But there were many, many benefits that accrued from neutralizing interleukin-1 beta. Mostly many inflammatory diseases got much better, and we published papers about that, which you can look up. If we look at people in a pre-specified but on-treatment analysis who responded to the neutralizing antibody by a greater than median lowering of C-reactive protein, there was an over 30% reduction in cardiovascular and all-cause mortality. Now, will all anti-inflammatory agents improve cardiovascular outcomes? Dr. Ritker conceived and led a study known as the Cardiovascular Inflammation Reduction Trial, or CIRT, which used low-dose weekly methotrexate, which has transformed rheumatologic diseases. And as you can see here, either with the primary endpoint or an expanded major adverse cardiovascular event endpoint, there was no benefit. But in this particular population of patients, there was also no anti-inflammatory effect. So if we contrast the positive CANTOS trial to the null cardiovascular inflammation reduction trial with methotrexate, we saw a reduction in the mediators that are associated with the NLRP3 inflammasome pathway, as shown here, which did not happen with methotrexate. So there's a scorecard here, which we published with Brendan Everett, one of our collaborators, a few years ago, showing that many things that can dampen inflammation prove beneficial in preclinical studies. Pilot clinical studies are often promising, but the outcome trials have been disappointing, except in the case of interleukin-1 beta, perhaps the icosapent ethyl, and very recently, we have expanded data supporting another anti-inflammatory therapy, colchicine, which in the COLCOT trial, in patients who were soon after an acute coronary syndrome, reduced events, and in LODOCO2 in individuals with stable coronary artery disease also had a striking reduction in recurrent events. So as we speak now, this is being recorded in the fall, COLCOT was positive in early patients post-ACS, LODOCO2 in patients with stable coronary artery disease, and there was a small Australian study looking in acute coronary syndromes that was null, but perhaps underpowered. With Hafid Etoufela and Alain Tegui at Parc in Paris, we wrote this review if you'd like to know what we're thinking about other anti-cytokine and immune therapies to reduce cardiovascular risk. But our research group is very focused on the NLRP3 inflammasome pathway that I've been speaking about. We can use inflammasome inhibitors very proximally, anti-IL-1 beta and IL-1 receptor antagonist, anti-IL-6 and anti-IL-6 receptor antibodies, and we hope that we'll have several more ways to mine this pathway and optimize patient outcomes. So I'm going to close by summoning up Sir Winston Churchill, who during a turning point in the great Second War said, "Now this is not the end, it is not even the beginning of the end, but it is perhaps the end of the beginning." And in January 2021, at the Journée Européenne de la Société Française de Cardiologie, I think that's where we are. We're at the end of the beginning of harnessing the 1.5 million papers on inflammation and atherosclerosis to give us a tool which will allow us to help our patients. Of course, it takes a big team to do this kind of work. And I've mentioned a number of these individuals during my talk. And we also had generous support from a number of granting organizations. Thank you very much for the opportunity of participating in this virtual Journée Européenne de Cardiologie. Et j'espère que la prochaine fois, on se verra face à face. Merci beaucoup.